In the previous video, I described glycolysis, the first step in all forms of cellular respiration. Now I'm going to describe the remaining steps of aerobic respiration. First, let's review the anatomy of a mitochondria where these steps occur. The mitochondrion is a double membrane structure with an outer membrane and an inner membrane. Between these two is an inner membrane space. In the center is a space known as the matrix. The inner membrane is folded to increase surface area, and these folds are known as cristae. At the end of glycolysis, the cell has two pyruvate molecules. These are carried into the mitochondria where they get altered by a multi-enzyme complex. This involves a decarboxylation. It's just a fancy word for removing a carbon. The removed carbon joins with oxygen to form a carbon dioxide molecule. A dehydrogenation also occurs, resulting in an NADH being formed. The pyruvate has now been converted into an acetyl group which binds to coenzyme A. We refer to this as acetyl-CoA. This diagram just shows you what happens to one pyruvate molecule, but remember there are two pyruvate formed from each glucose molecule. So at the end of this step, the cell has made 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. Coenzyme A transports its acetyl group to the next step known as the citric acid cycle or the Krebs cycle, which occurs in the matrix of the mitochondria. It may look complicated, but I'm going to break it down here, and you do not need to remember these individual steps. Coenzyme A hands off the acetyl group to the cycle and returns to the previous step to shuttle more acetyl groups. The 2-carbon acetyl group gets attached to a molecule called oxaloacetate and forms a 6-carbon molecule called citrate. A series of reactions then oxidizes the acetyl group to harvest all its usable energy. The citrate is transformed in a series of redox reactions, 2 decarboxylations, and a substrate-level phosphorylation. The final reactions restore the oxaloacetate so it can then accept another acetyl group, and the cycle repeats. Since there are 2 acetyl groups coming from that original glucose molecule, the cycle turns twice for each glucose molecule that goes through aerobic respiration. Therefore, the yield for 1 glucose, or 2 turns of the cycle, is 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. At this point, the glucose molecule has been completely oxidized. All 6 carbons are now in the form of carbon dioxide, and all the energy from that glucose molecule has been put into our taxicabs or into ATP. After glycolysis, the prep step, and the citric acid cycle, the cell has a net yield of 4 ATP, 2 from glycolysis and 2 from the Krebs cycle. There is a total of 10 NADH, 2 from glycolysis, 2 from the prep step, and 6 from the Krebs cycle. And there are 2 FADH2 from the Krebs cycle. The last step is the electron transport chain and chemiosmosis. This is where NADH and FADH2 drop off their high energy electrons. They drop off these electron passengers so they can go back and get more from the previous steps as glucose and other organic molecules are continuously fed into this pathway. The electron transport chain is a series of electron carriers and pumps that are embedded in the cristae of the mitochondria. NADH drops off its electrons to the first member of the chain, and FADH2 drops off its electrons to the second member. The electrons are then passed from one carrier to the next, and as they pass, the energy they contain is used to pump hydrogen ions out of the matrix into the inner membrane space. These three proteins are the pumps. This creates a concentration gradient with a high concentration of protons in the inner membrane and a low concentration in the matrix. You learned about concentration gradients previously. Once the electron transport chain has squeezed all the energy it can possibly get out of these electrons, the electrons are accepted by oxygen and two hydrogen ions to form water. The different types of cellular respiration are based on the type of final electron acceptor. Cells cannot tolerate free electrons floating around. In aerobic respiration, the final electron acceptor is oxygen. Aerobic literally means with air. Anaerobic forms of respiration do not use oxygen and will be described in Part 3. Adjacent to the electron transport chain on the cristae is the enzyme ATP synthase. Here we have an electron transport chain pumping hydrogen ions out into the inner membrane space, and then the ATP synthase right here allows those hydrogens to flow by diffusion back into the matrix, and that flow turns the enzyme like a water wheel, and that mechanical energy generates ATP. This is chemiosmosis, also known as oxidative phosphorylation, our second method of making ATP. Let's review the energy yield for this pathway. In glycolysis, a glucose molecule was oxidized into pyruvate, and this generated 2 ATP by substrate level phosphorylation, and 2 NADH were formed. The pyruvate was oxidized into acetyl-CoA, and this generated 2 NADH. 2 acetyl groups were oxidized in the citric acid cycle, and this generated 2 ATP by substrate level phosphorylation. 6 NADH were formed, and 2 FADH2. A total of 10 NADH and 2 FADH2 were then shuttled into the electron transport chain to produce ATP by oxidative phosphorylation, which is the electron transport chain in chemiosmosis. The ATP gained from this step varies depending on the organism. Most textbooks will list 30 to 34 ATP. So the total ATP yield is often stated as 36 ATP in textbooks. But current estimates are 29 to 30 ATP per glucose due to leaky membranes and the cost of moving pyruvate and ADP into the mitochondria. The whole process is about 32% efficient. In biological terms, this is high. Also, keep in mind that some energy is lost as heat. When I describe anaerobic respiration in the next video, you will see how much more efficient aerobic respiration is by comparison. Cells break down other organic molecules besides glucose. For example, the proteins and triglycerides humans eat can be used to make ATP. Proteins are broken down into amino acids. The amino acid basic structure has a nitrogen, and the respiration pathway is not equipped to handle nitrogen. So, that nitrogen in the amino group is removed in a step called deamination. The remainder of the amino acid can then be converted into a molecule that can either enter glycolysis or can be converted into a substrate for the citric acid cycle. It all depends on the variable R group and how many carbons, hydrogens, and oxygens are in the molecule. Triglycerides are broken into glycerol and fatty acid chains. The glycerol is a 3-carbon molecule the cell can easily slip into the glycolysis pathway. The fatty acids are broken apart in a process called beta oxidation, which cleaves the chain into 2-carbon acetyl groups. A 6-carbon fatty acid chain will actually yield 20% more ATP than a 6-carbon glucose because the glucose is split into 2 acetyl groups, while the 6-carbon fatty acid chain is chopped into 3 acetyl groups, turning the citric acid cycle 3 times instead of only twice. Even nucleic acids can be used in this pathway to make ATP. Please watch Part 3 for a description of anaerobic respiration. Thank you for watching.